right, so the messenger RNA has already gone through the transcription process. That was where it went and copied one side of the DNA. And now it has left the nucleus. And it has come outside of the nucleus into the cytoplasm of the cell. It's still inside of the same exact cell, but now it's gone to a different organelle, and that organelle is called a ribosome. And the ribosomes are the protein factories in the cell. It's where the proteins are made. So basically what we have are some strands of messenger RNA here. I took them off the string just to make them easier to work with. So this was the original one that we made in the last video. And then you see we have 4 separate messenger RNA strands here. Each of these is coding for one gene. Okay? So that one gene is going to build a protein. So each of these is going to build a different protein. So we're going to start by taking one strand of messenger RNA and running it, literally threading it through a ribosome. Only one strand goes through at a time. And as it is read, it's read in groups of 3 bases. This is how the code starts. So AUG is read as a code, and actually it's called a codon. Each group of 3 bases is called a codon, and the codons are what reveal the code. So the next 3 are CUG, so there's another codon. Then we have ACU, there's another codon, and UAA, there's another codon. All right, so the second strand, let's do it out real quick. AUG, UCC, UAU, and UAA. The next one, AUG, GGA, UUC, and UAA. And the last one is AUG, AAC, CGC, UAA. You might notice these 4 different strands coming from 4 completely different genes have a few things in common. One is not something they would normally have in common. They're all the same length, and that's just for ease of showing you what's going on. But if you look really closely, you'll see that each of them start with a codon AUG. And guess what that codon is called? The start codon. So that makes it really easy to remember. When the ribosome sees AUG, that means start building a protein. So it might see a whole long strand of mRNA stuff coming through, mRNA, but when it sees AUG, boom, start. Now it's time to start building a protein. That's important. You don't want it just starting anywhere in the chain. It needs to start here. Just like if you were building Hogwarts, you wouldn't start by building the bathrooms, right? You'd probably start by building the outside, build the foundation. Then eventually you'd get to the inside and start building that. You would not start in some weird random place. You'd start at the beginning. That's how proteins are built. They start at the beginning, at the start codon, which is always AUG. Then we have 2 other codons here. They're all random. None of these are the same. But when we get to the bottom, we see they all end in UAA. Right? This is called a stop codon. Easy to remember. And guess what that function is? To stop the protein from being built. Because once it's done, the ribosome needs to know this is the end, we're done with this protein, we can release it and we're finished. Now there are 3 different stop codons that are in your lesson plan. I'm just using UAA just to make it easy for us. Alright, so we're going to try this out and see how this works. Messenger RNA, again, is going to be threaded through the ribosome. The ribosome is reading each codon. Right, so the first one that it comes to is AUG. And guess what? We've got to match pairs yet again, guys. So we had our original DNA strand that mRNA matched base pairs off of and brought to the ribosome. Now, to get our amino acids, which are the building blocks of the proteins, we have to match again. It's the last time, I promise. All right, so this is transfer RNA, tRNA. And its job is to transfer the code and transfer it into a amino acid, right? It's going to go get it. So AUG would match to UAC, and guess what that is? Start. Boom. So let's put it up here. We know what we are about to start building. Maybe it'll fit. We're going to start building a protein. Okay. Now let's go to our next codon, CUG. Well, we should know that the transfer RNA, the tRNA for this one, should have what we call an anticodon. These here are the anticodons. It's the match. Should have GAC. So that's exactly what happens. Another transfer RNA comes in that matches GAC. Boom. And it's now we know we're started building, so now transfer RNA has to actually go get that amino acid. It's floating out in the cytoplasm of the cell. I'm going to hand that off to Sophia, my assistant. She's going to be my transfer RNA, and she's going to go find the amino acid that matches. And look, she's brought it to the ribosome. Perfect. The ribosome sees it. It actually will take it off of the transfer RNA, and the transfer RNA will go back floating around, waiting for its next assignment. So put that in place for me, transfer RNA. Next codon that comes down the chain is ACU. Well, we know we've got to match that with UGA. And here it is. It's coding for this amino acid, THR. These are all abbreviations for different amino acids. We'll talk about them here in just a minute. So this one's going to code for this. So transfer RNA is going to leave the ribosome. It knows exactly what it needs to grab. It goes down, finds it in the cytoplasm somewhere, and it's going to bring it back to the ribosome. All right, and here it is. This matches THR, THR, perfect. The ribosome is going to break it off of there, and then the transfer RNA goes floating back out into the universe of the cell, and we'll put it in place. Look here. Our amino acids are slowly starting to form a protein that we may recognize so far. Now, is it the full protein? No, it's a part of a protein. We're getting there. Okay, now the next one is UAA, which codes to AUU. And guess what, guys? That's our stop codon. Boom. Gryffindor is built. The entire protein of Gryffindor is built. Now, is that everything? No, it's not the entire organism, okay? It's not the entire organism. It's a part of it. It's one trait of our organism. The gene codes for one trait. The messenger RNA goes to the DNA, finds the one gene that it needs to get the protein for, copies that off of the normal DNA in the cell, brings it to the ribosome, and the transfer RNA goes and gets the amino acid that we need to build that protein. So now we've built one. Let's go back and do the next one. All right. We already know AUG is going to be a start. So here we go. We're going to start building another protein. The next amino acid code is UCC. So that's going to match to AGG. That's coding for SER. So transfer RNA runs out into the cytoplasm. It finds the amino acid that matches SER. Here it is. SER, SER. Put it in place. And as soon as the ribosome takes the amino acid off, the transfer, whoopsie, the transfer RNA goes back into the cytoplasm. The next one down here is UAU. So that's AUA, codes for this amino acid. Transfer RNA goes from the ribosome, runs out into the cytoplasm, finds the TYR amino acid, brings it back to the ribosome. The ribosome takes it off, pieces it together, and off the transfer RNA goes. Okay, the next one is UAA. We already know that that's AUU. That is our stop codon. So guess what? We are done building the protein for Ravenclaw. All right? Again, is our organism fully built? No. We've built two traits from two different genes so far. Okay, next up, AUG. Again, that is our start codon. We're about to build another one. And here we go. GGA. GGA codes for CCU. GLY amino acid. Transfer RNA runs to go find it, grabs it, there it is. The ribosome removes the amino acid, releases the tRNA back into the cytoplasm in case it needs to run out and grab another one of those, and another piece is in place. Next up, it's a little rickety.
Yeah.
There you go. Okay, next up is UCC. Whoops.
Whoopsie.
I messed up somewhere, didn't I?
Yeah, you did.
UUC should be, uh-oh, these went backwards. Haha, see, we have a problem here. These got put on backwards. This should actually be AAG. All right, let me fix it real quick. Now, in the cell, this would be a problem. A big problem that we'll talk about next week. But for now, we don't need any mutations. So, AAG. There we go. All right, so AAG codes for the amino acid PHE. Transfer RNA runs out into the cytoplasm, finds that amino acid, brings it back to the ribosome. It matches, the ribosome pops that amino acid off, adds it to that protein chain. Transfer RNA goes back into the cytoplasm. The next one is UAA again. Boom, 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 our stop codon. And that's the end of that one. And look, we have built a complete protein for Ravenclaw. Okay, organism still isn't done, right? So now we have another strand of messenger RNA. Let's do it, and then it looks like we might be done with our organism. Okay, so we have our start codon. Here we go. AAC should go with UUG, coding for the amino acid ASN. Transfer RNA runs out into the cytoplasm, grabs it, comes back to the ribosome. Ribosome takes it off, puts it into the chain, and transfer RNA heads back out of the cytoplasm until it's needed again. Next up, CGC reads for GCG, ARG amino acid, transfer RNA. It's his job to head out to the cytoplasm, bring it back to the ribosome. Here it is, matches up, transfer RNA back out into the cytoplasm, ribosome removes that amino acid, puts it in line with the protein, and then we get a UAA, which is an AUU. That is our stop, and look at that. We've built an organism with 4 different traits, and that is exactly how it happens, guys. So, to sum it up, we have that DNA in our original cell that has the code. Messenger RNA, when it needs to build a protein, comes into the original cell, copies one side of the DNA, except instead of using T, it uses U. It then is only one strand. It comes out of the nucleus into the cytoplasm of the cell and finds a ribosome. That's the protein factory. That's where it's going to build it. The messenger RNA strings itself through the ribosome, and the ribosome starts reading it in codons, right, which are groups of 3 bases. AUG is always the start codon, telling that ribosome it's time to begin building a new protein. And then for each codon below it, we will have an anti-codon on a transfer RNA that will match up. Okay? That codon, the anti-codon, will run out and grab whichever amino acid is needed in the chain, in order. Very important that it's in order. Okay? It will bring that amino acid back to the ribosome. The ribosome will break that amino acid off, stick it onto the chain, and let transfer RNA go about its business to go grab another one as soon as it's needed. This is happening very fast. Once we reach the stop codon, the end of that chain is snapped off and that entire protein is built. And that is a piece of a trait to be built from that gene. It could be hair, it could be heart cell, it could be a muscle cell, it could be a bone cell, it could be any type of trait that you might have. And all of them all together, that's going to build your entire organism.